In laboratories, hospitals, and classrooms all over Texas, today's innovators are blazing a trail into the future. The Academy of Medicine, Engineering, and Science of Texas presents the Edith and Peter O'Donnell Awards to individuals who are keeping Texas on the leading edge.
The peaks are very clear and detectable.
Can we use this to back it up?
When other couples are discussing ways to save on the grocery bill, this husband and wife are talking about research that can save lives.
So as a validation, I say, what are you using as a negative control?
At Houston's MD Anderson Cancer Center, Wadi Arap and Renata Pasqualini are fighting a deadly disease, one that hit close to home.
Renata had her mom with breast cancer. I had my father who has colon cancer and is getting chemotherapy for the past year. So it is incredibly humbling not to be able to help.
What could help is a discovery Wadi and Renata made. It's something called a zip code system.
The molecular mapping of the human body in the sense of finding addresses in specific tissues, specific diseases to which you can deliver payloads at will.
The discovery is based on the fact that peptides in the blood vessels in cancerous tumors are different from peptides in normal blood vessels. Wadi and Renata believe that if you can identify that difference, or zip code, you can design a peptide that when injected into the bloodstream will go directly to the tumor. The potential benefit comes when you attach an agent to the peptide that can treat the cancer.
You can use any form of chemotherapy if you like. You can use regulatory proteins. You can use imaging agents or radioactive agents that then you can see and at the same time treat the tumor.
This targeted treatment could greatly reduce the side effects of chemotherapy, as the chemicals would not be delivered to healthy tissues.
So this particular drug was discovered in a human screening.
Renata and Wadi have developed this zip code drug. It worked well in animals, and now the FDA has approved its use in clinical trials in human cancer patients.
As a first-in-human study, I think we'll be able to bring some very exciting proof of concept to the idea.
Renata and Wadi have developed another zip code drug that appears to destroy fat tissue. Many in the medical community are impressed by the couple's work.
I think it can be easy to overlook sometimes the amount of both scientific and clinical effort to translate that from an idea in just a decade all the way to patient applications. I think it's remarkable what they have been able to achieve.
Wadi and Renata shared their lives and the same positive attitude about beating cancer.
If you're doing something new, is even more resistant to acceptance in general. But if you're confident and you keep at it, ultimately it works.
People that go with the flow, it's very unlikely that they will enact any difference.
Here, all you need to do is walk around 2 minutes and you see in front of you the realities of cancer, and that's a huge motivation. It's a long road, it's not easy, but it's not impossible.
If we're willing to potentially statically map the instructions to these ALUs, we can encode where to send things.
In whiteboard discussions, UT computer science professors Steve Keckler and Doug Berger made discoveries that ruffled some feathers. They challenged the way computer microprocessors are designed.
The prototype chip that was designed here in this building had 170 million transistors. It was among the most complex chips and system efforts done in academia, period.
As children, Doug and Steve were both fascinated by science. So it's not surprising that in college, they both specialized in computer architecture.
We first met, I think, in 1993 at a computer architecture conference when we were graduate students, and kept seeing each other over and over again, and our lives kept intersecting in various ways.
Doug and Steve decided to team up. They started the TRIPS lab at UT, a project aimed at discovering ways to improve the performance of computers.
We're simulating, I think, 48 entries on T-Flex.
That's right.
Now,
That's
it'll
right.
still take a little bit of a performance hit
Doug
on
and
some
Steve
workloads.
made a controversial discovery. In 2000, they published a paper that said computers would reach their performance limits. Doug and Steve warned the tech industry it needed to stop designing microprocessors for computers that had large centralized components. The wires and switches on those components were becoming overloaded.
If you centralize a structure in the real world or in silicon, it's more efficient for things to communicate within it, but as it grows, it gets more and more unwieldy. So imagine airports. If you tried to centralize all airports in a single location.
So what were we able to measure in terms of the temperature and the power and the correlation between the two?
So the correlation between
The TRIPS lab allowed them to design and build a prototype of a microprocessor that had much higher peak performance than the chips the industry was using. The TRIPS chip featured multiple copies of components. They were connected to each other so that they could share the workload. The results Steve and Doug produced sent chip makers back to the drawing board.
Not only did it inspire people to look at radically different new microarchitectures for the future, but it also inspired the way we design compatible x86 microprocessors. It really did shake people up a bit.
Now Steve and Doug are making the TRIPS chip even better.
We're now on our third generation microarchitecture and have some very, very compelling results. I just want to see this technology widely deployed. I mean, it's everyone thinks their baby is beautiful, right?
This has been a very rewarding period of my life to be working with Doug and the students that we've recruited. It's just been a fantastic team.
You're only using a 1-inch optic at the end.
Which one, right here?
Yeah.
Yes, that's the small one.
Is that a disadvantage?
In labs at Texas A&M, Paul Kramer leads a group of young scientists on a mission to unlock the biological mystery of how germs attack cells in the body.
That's what we try to study and understand, the underlying biophysics of membranes and how these motions occur, and what kinds of properties, chemical properties, one can change to actually change how the infection would occur.
It's knowledge that could lead to the development of drugs to fight dangerous illnesses like influenza. It's the kind of work Paul was born and raised to do.
My father is actually an organic chemist and my mom's a teacher, and I was raised in an environment with a lot of inquiry.
Now, it's Paul's understanding of the relationship between biology, chemistry, physics, and technology that has given the Kramer Group the tools to perform groundbreaking experiments. They were the first to coat microfluidic and lab-on-a-chip platforms with supported membranes. By coating the surface of the chip with lipid bilayers that mimic cell membranes, Paul's group can study how viruses, toxins, and proteins can interact with cells.
No salt, 500 millimolar of which salt? Okay.
Paul also developed processes using nonlinear spectroscopy and microfluidics to study cell interactions. Illness is spread when the precise conditions occur in cells that allow germs to attack. The tools Paul created allow his team to determine what those conditions are with amazing efficiency.
You can probe things as a function of temperature, as a function of concentration, pH, and perhaps control them all simultaneously with just hundreds of nanoliters or a few microliters of material to make hundreds of measurements simultaneously. Then you can actually hone in on what exactly those functional processes are.
Paul Kramer's work should help make a difference for hip and knee replacement patients. His investigations have led to a better understanding of the root causes of why the body might reject implants. His work also led to a better understanding of protein aggregation, which lies at the heart of diseases like Parkinson's and Alzheimer's. Paul is helping put Texas A&M on the cutting edge.
We're very fortunate to have in Paul someone who is able to bring together physics, chemistry, and biology. And in a very real sense, Paul's work is laying the foundations for future bioscience and technology.
We still have plenty of new territory to plow on almost a daily basis. Just about any question that we can ask, if we can understand the molecular level details of processes, they're often very helpful to the community at large, or at least that's our goal, is to make them helpful to the community at large.
How's this machine coming along?
Coming along very nicely. We're in the process.
S.V. Srinivasan is on a constant quest to push technology to its limits. His hope is that the machines and processes he designed for molecular imprints in Austin will quench consumers' thirst for gadgets and computers that can do more, do it faster, and take up less space.
So the pitch of the discrete track here is what roughly?
SV's machines print nano-resolution features onto computer hard disks and semiconductor wafers that are the brains of today's electronic devices. Features that are 2,000 times thinner than a human hair.
You can make very high density storage devices, which helps people manage their data.
One of the machines SV's team developed works by imprinting nanoscale ridges on the surface of computer disk drives. The ridges separate magnetic bits of data on the disks so their magnetic fields won't interfere with one another. The manufacturing process involves capturing a photosensitive liquid inside a mold and hardening it with ultraviolet light. The result is a precise pattern of ridges, neatly separating the sensitive magnetic bits.
We are able to basically pattern matter at the nanoscale, materials at the nanoscale, at a very reasonable cost, much lower cost than anybody's ever done before.
SV's breakthroughs came from a collaboration that began in 1996 with Dr. Grant Wilson, a chemist at the University of Texas. Wilson had developed an imprinting process that used photosensitive materials. He just couldn't figure out how to remove the mold without damaging the extraordinarily fine features.
You need to remove the mold without rotating it on the vertical axis or translating it in the plane of the image, even a few nanometers, or you'll smear off the image.
For help, Dr. Wilson turned to SV, then a new mechanical engineering professor at UT, who specialized in building precision machinery. SV's team quickly built a machine that could reliably remove the mold from the substrate without damaging the fine features. SV's team didn't stop there. They developed other innovations that made the concept viable.
We developed this ink jetting technique, a very precise picoliter volume ink jetting technique, where we can put down the liquid as a function of what is on the device, which is the mold.
The machine worked so well that SV founded Molecular Imprints in 2001 to bring his technology to market. SV's company could someday produce nanoparticles that fight diseases, or LED lighting that's more powerful and energy efficient.
We're now in the phase of figuring out how to get into volume manufacturing and really ramp up, because that's when you can say you've had societal impact, when people are buying stuff that requires your process. So we're pretty close to that, and it's very exciting.
Congratulations to these 6 rising stars of research and innovation in the Lone Star State.